Welcome back, welcome back, welcome back to science. All right, we're looking at the adaptive immune system. We just looked at the innate. Now we're looking at the adaptive. The adaptive is going to be specialized. This only happens in certain higher order animals. So, not all animals have this. Mammals have this. This is considered to be the third line of defense. This is our ability as a really high level organism to be able to identify something and develop a response to it that is really specific. And our cells are able to remember what that thing is. So if they see it again, they can have a really driven immune response. So the cells have to be able to distinguish self from that's part of the body from non-self. And they do that with cell markers called antigens. On the outside of cells, oftentimes there are different kinds of pieces, whether it's proteins or glycoproteins or glycolipids, that act as kind of like a name tag. That specialized region allows for the cells to identify self from non-self. The adaptive immune response is going to have two different pathways for attacking things that are non self. So, adaptive defenses are divided into the cellular um, immune response, called cell mediated immune response, and also the humoral response. Humoral, let me give you a way to remember humoral. Back in the old days, the fluids of the body had to be balanced. And I say old days, go back a couple hundred years, and this was thought to be the case that we had to have enough yellow, enough red, enough brown, enough green. Um, so that's urine, it's blood, it's feces, it's mucus. If we had one that was in too large of a proportion, we would try to fix it, and that's part of what bloodletting was about, cutting and trying to bleed out because we had too much blood. Um, Not necessarily good science, but those were the humors of the body. And so the fluids of the body is where one of them are. One of those is really focused, and the other one is focused on a specific type of cell in the cellular location. So these are going to be the cell mediated immune response is going to be the T cells, and the humoral response are going to be the B cells. We'll get into that. Here's our overview of line of defenses. Here as a pathogen begins to enter the body. It comes to the first line of defense, which is our skin and our mucous membranes. It prevents most things from getting in. An unsung hero. If the pathogen gets past that, the next thing is the second line of defense, where we have chemical barriers like the enzymes that are in our skin, the pH of our skin, salt, interferons, defensins, collectins, and the complement system, along with some cells like the natural killer cells, inflammation, phagocytosis, and even fever to help with the second line of defense. The third line of defense, this is our final line. And this is going to be that adaptive immunity that is so specific that it is like a sniper, is the better way to look at it. The way it's going to individualize something and remove it. So, the antigen, the part that's identifying as non self. Antigens can be these different things. That is, they can be proteins, they can be polysaccharides, they can be glycoproteins, they can be glycolipids. Ultimately, they're like a chemical name tag. Once again, cells communicate chemically, and these are chemical structures on that cell surface that you're going to find that's so unique to them. Haptens are something that's are antigen-like. They're small molecules that are not exactly antigenic, but they're going to have larger things that combine with them that can then develop an immune response. We'll see haptens in specialized cases, but that's not going to be the focus of where we are today. All right, so lymphocyte origins. Remember that there are the B lymphocytes and T lymphocytes. All of these are going to start their production in red bone marrow. So, for the bulk anyway. Some of them are going to then move to the thymus where they're going to have final maturation occur. Remember the hormone thymosin that's produced by the thymus, which will trigger the maturation of T cells that can then be released to do their job. So they're going to specialize in the thymus. They're going to make up most of our lymphocytes. There's 70 to 80% of them are going to be the T cells. Some T cells are going to settle in lymphatic organs, like as we talked about, like the spleen, how they're going to be a lot in the white pulp. But there are also some that's going to be in the red pulp. They're also going to be found in areas like our lymph nodes and those encapsulated region, our thoracic duct, the white pulp of the spleen. These are all areas where we can find these T lymphocytes. Now, B lymphocytes are going to be named like where the T is going to take the T of thymus and T lymphocytes. That makes sense because they're thymus derived. Due to some early research, the bursa of fabricus in chickens was where we found and has really started the studying process of the B lymphocytes. And that's where they got their name for B lymphocytes. So after the red bone marrow has made those lymphocytes, they're going to be released. These are going to be very abundant also in our lymphatic organs. They're going to be found in our lymph node, spleen, bone marrow, and even our intestinal lining. The intestinal lining, we'll get to that later on why that's important. So this is showing the B cells being generated from solely the red bone marrow. And we have the T cells, which are started in the bone marrow, and they have maturation in the thymus. Then they're going to be released, and you can see them starting to accumulate in a lymph node doing their job. All right, let's start talking about T cells. T cells are not activated until they have something that says, like, it's time to go. And there's going to be a type of cell that's going to phagocytize and eat something. And this is going to be an antigen presenting cell. The antigen presenting cell, just like what you would think, is going to eat something, phagocytize, and it's going to take part of that phagocytized thing and it's going to show that to another cell. So it eats, it takes a piece of the scrap of its cellular marker, and it's then going to show that to another specialized cell. Now let's talk a little bit about the mechanism of doing this. What I'm using to kind of as my hand to hold out that piece, the hand would represent something that's called a major histocompatibility complex. A major histocompatibility complex holds out the antigen of that microbe, the part that's the cellular marker of essentially the antigen of that microbe. It's going to show that to another cell. These specialized T cells are then going to develop a response. So a T cells are going to identify that. And so now that we've identified that structure, we now have specialized T cell reaction. And T cells are going to start talking with their language of something called cytokines. And cytokines are going to enhance the cellular response. Let's identify some specialized T cells. The first one we're going to identify is the helper T cell. The helper T cell is going to activate other cells, including B cells. HIV, we'll talk about it a little bit later. It's going to target helper T cells, and since helper T cells can't respond because they're essentially muted, the B cells are not ever really turned on. So you can't have a whole area of our adaptive immunity to go. And so this is going to cause an autoimmune disorder where you're going to have the lack of production of an immune response. So they're going to be immunosuppressed as a result of the lack of the T cells because the virus is targeting the T cells, the helper T cells. All right, cytotoxic T cells are going to attack virally infected or cancer cells. Now they're going to use perforins to do this. And memory T cells, this is my favorite part. The memory T cells are able to make a long-term memory of how to defeat that specific antigen. So if it ever sees it again, it already has all the tools already hammered out, ready to go, and it just turns on the machines to make that. Instead of like we need to machine out the right tool and then develop a response. Instead, it's like we just need to make copies of it. Go, 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 go. These are examples of cytokines. We have colony stimulating factors that stimulate bone marrow to produce lymphocytes. We have interferons that block viral replication, stimulate macrophages to engulf viruses, stimulate B cells to produce antibodies, and attack cancer cells. The interleukins are going to control the lymphocyte differentiation and proliferation, and tumor necrosis factors stops tumor growth, release growth factors and causes fevers that accompany bacterial infection, and stimulates the lymphocyte proliferation. All right, B cells, also called the humoral immune response, because the B cells are going to release antibodies. B cells are going to be activated by the helper T cells. So once you have the cytokines from the helper T cells, this is going to tell the B cells, like, hey, it's time to go. So now we're going to develop an immune response with our B cells as well. Our B lymphocytes are turned on. So the B cells are going to differentiate into both plasma cells, and plasma cells are going to release antibodies. Antibodies are going to actually leave those B cells, and they're going to go out into the humerus of the body, the fluids of the body. They're going to tag whatever kind of microbe identifying the antigen, and then there's going to be an ushering back for phagocytic reaction to occur with a type of macrophage. So they're going to use these specialized antibodies. Antibodies are also called immunoglobulins, and there's going to be 5 different kinds of immunoglobulins. So since antibodies are carried in the blood or the body fluid to the infection site, the type of response is called the humoral immune response or antibody-mediated immune response. So here is the antigen. You can see the receptor-antigen combination, and here is the receptor region. When you have that combination, this is going to have the activation of the B cell, and this is by the cytokines telling it, hey, it's time to go. So now you're going to have clones of these specialized, because it's going to start making proteins, and it's going to make receptors that are going to be specialized with those tips. And it's also going to start having the plasma cells release antibodies out from those B cells. These are the antibody secreting cells. Here we have memory cells, and as you would guess it, this is long-term storage of how to defeat that. So these are all in the clone of B cells that exist, and it's it's performing long-term memory of this with those memory cells that are going to remain dormant, along with the humoral response with those B cells. So here is the activities, the T cell activities, the antigen-bearing agents by the antigen-presenting cells enters the tissue. We have the major histocompatibility complex that's then going to become specialized. We're going to have the antigens that's going to, from the digestive antigen-bearing agents, are displayed with that major histocompatibility complex. The helper T cells become activated when it encounters a displayed antigen. Then you're going to have the activating of the T cells are going to release the cytokines that are going to cause the B cells that are going to become tooled to be now exact at releasing the right antibodies that are going to be having those plasma cells releasing that into the system, as you can see on step 4 up here as well. Now you're going to have some of the newly formed B cells give rise to the differentiated into the antibody screening plasma cells. All of these are going to be contributing to that immune response. So let's look at antibodies. So they're globular proteins, meaning that it's going to be this quaternary structure, so multiple structures put together, really specified. These structures that make up the gamma globulin fraction of plasma proteins make up the immunoglobulins. They're going to have a Y-like shape. They're going to be composed of four amino acids, two heavy chain, two light chain. Of those amino acids, of those antibodies, there's going to be five different categories of those immunoglobulins. And so if you think of GAMED, GAMDE, those are going to be the sequence and the frequency in order. So IgG is the most common of the antibodies. 80% of them are going to be IgG. This is going to be produced, they're going to act on bacteria, viruses, and toxins. IgA is going to be found in exocrine gland secretions. You're going to have IgM, that's a 6%, also in response to things like food and bacteria. IgD is going to be found on the surface of B cells. This is going to be common in infants. And IgE, this is also going to be from exocrine gland secretions. If you do an assay of blood and find which antibodies are going to be there, those immunoglobulins, it can also help you narrow down what kind of infection you're dealing with. So this is an antibody. This is the heavy chain that's on the center region, and here's the light chain. This is the antigen binding site where the antigen is going to fit there perfectly for whatever it is that is trying to identify. This is the variable region, meaning that it can be tooled like a multi-tool. You can put on a different head, tool a specialized head for doing a specialized task. And then it remembers that, and it's going to make a lot of those. All right, so antibody actions. Antibodies respond to antigens in three different ways. So if you remember when we talked about blood and we talked about if you're given the wrong blood type, let's say you give type AB to a type O blood, that person is going to have agglutination. This is going to be the clumping of antigens. This is going to make phagocytosis easier. And so you're trying to identify those problems and you're going to cause them to clump. Notice that I didn't say clot. That's a different thing from the science standpoint. Another thing that can happen as a result of antibodies is precipitation. This is going to make antigens insoluble. It's going to allow you to form a really precise attack phagocytically. And the next is neutralization. This is taking a toxic portion of an antigen and making it harmless, kind of taking the teeth out of that antigen while you're developing your immune response. So, activation of complement. The more effective against antigens than direct attack. So, this occurs with certain IgM and IgG antibodies. Actions of activation complement proteins include opsonization, chemotaxis, lysis, and neutralization. Opsonization is the coat the antigen antibody complex. There's not a good image of that. Sometimes there are. Chemotaxis. This is going to be a positive response to be able to go through something. This is going to attract specifically the macrophages and the neutrophils. Once again, the granulocytes. We're going to have lysis, where we're ripping, opening, and rupturing the cells of the pathogen. And we have neutralization, where we're essentially making it harmless by either putting an antibody on it to be marked or to enable, disassemble the thing that is causing the problem. So the stimulation of local change, inflammation, and to help the spread of infection, IgE on mast cells. Remember mast cells that we talked about? These are the histamine producers. The histamines are going to cause the leakiness of vessels, which is going to cause secretions to occur, and it's also going to cause edema, where you're going to decrease the effect of that microbe. Right. Immunotherapy. This is something that became well known to every household, I feel like, during the COVID years. So, immunotherapy makes the immune system complements to fight disease. So, this is the humoral response by antibodies and the cellular immune response by the cytokines. Monoclonal antibodies are a word you may have thrown around a little bit. You may know what they are, may not. But this is going to be a B cell product, and it's going to be the product of a single type of antibody, the monoclonal, and they're going to fuse with cancer cells to produce a cell type that produces one type of antibody, and they just keep dividing. So we've made a specialized antibody producer. This is used in research, agriculture, pregnancy tests, detecting new or reoccurring cancers, deriving medication specific to tumors. Another type of immunotherapy are the cytokines. This can now be produced in large quantity due to recombinant DNA and monoclonal antibody techniques. This is used to treat things like leukemia, multiple sclerosis, hepatitis, genital warts, melanoma, and even kidney cancer. All right, guys, this is going to end this subsection. I look forward to seeing you soon. So long.